Welcome to another lecture by Medical Medics, Immunology Chapter 4, Adaptive Immunity. In this lecture, we are diving into adaptive immunity, the components involved, the activation of the adaptive immunity. We will talk about specific cells like the T cells and B cells, and also talk about antibodies. We will talk about types of adaptive immune responses, immunological memory, the clinical relevance here, and end with a summary. Now, adaptive immunity is the body's specialized immune response that targets specific pathogens. Key features here include specificity, so it recognizes and attacks specific antigens. Memory is another key feature, so it remembers pathogens for faster responses in the future. Furthermore, slower initial response. It can take days to develop this response, but the response is precise and long-lasting. And you can almost consider the opposite when you're talking about innate immunity. So it's non-specific, it doesn't have memory, and it has a rapid initial response when we talk about innate immunity. Main components of the adaptive immune system is T lymphocytes. So we differentiate between helper T cells, CD4, and cytotoxic T cells, CD8. We have B lymphocytes, or B cells. These are important for producing antibodies. We have molecules like antigens and antibodies. So antigens are specific molecules that trigger an adaptive immune response. Antibodies are specialized proteins that neutralize pathogens. So again, to repeat, we have some pathogen entering our body. Let's say it's a bacteria. We have our first innate response to this. So we have neutrophils arriving. We then have macrophages coming, engulfing this bacteria, taking its antigen and presenting it on the surface of the macrophage. And this then triggers or makes a connection with our adaptive immune system. For example, a T cell will come and recognize this and then initiate a cascade of other events. One of these events is recruiting B cells. These B cells activate and produce antibodies. These antibodies are very specific proteins, very specialized type of proteins. And these antibodies will always remember to specifically target that antigen. And the B cells will multiply into a lot of antibodies. So it's mass-producing antibodies, specifically targeting this antigen. So the next time that same bacteria or a different type of bacteria with the same properties enter our body, we already have a strong defense immediately responding to it. So when antigen is presented, so we talked about just now macrophages, for example, but we also have dendritic cells that are antigen-presenting cells. They will capture antigens and present them to T cells in the lymph nodes. We then have helper T cells that release signals to coordinate the immune responses. We have cytotoxic T cells that may arrive, if told so, and come and destroy these infected cells. Another key feature of the helper T cell is to activate B cells to produce these antibodies. Now, T cells in adaptive immunity. So again, we have helper T cells, often referred to as CD4+. These help coordinate immune responses by releasing cytokines. They help activate B cells and cytotoxic T cells. The cytotoxic T cells, also referred to as CD8+, destroy virus-infected and cancerous cells by inducing apoptosis. Apoptosis is programmed cell death. And it's kind of suggestive by the name cytotoxic. Then we have regulatory T cells. These suppress excessive immune responses to maintain balance. So from the previous lectures, we now added some new concepts like apoptosis, connecting it to cytotoxic T cells. We also introduced regulatory T cells, which help suppress excessive immune responses. So we have checks and balances. Now, we also have B cells. When B cells are activated by the help of helper T cells and exposure to antigen, they will differentiate into plasma cells. These plasma cells are also so-called antibody factories and into memory B cells. So antibodies have a Y-shaped structure, and it's a protein with variable regions that bind specific antigens. Functions here include neutralizing pathogens, opsonize pathogens, meaning marking them for destruction, and activate complement proteins. We have an in-depth lecture in our anatomy classes, actually, on the immune system, and we will attach a lecture slide where we go into more specific details on the structure of the antibody here in this lecture. Now let's just dive a little bit deeper into the B cells. So let's talk about antibodies and their functions. Now we have a detailed course on immunology where we talk about all of these different cells in great detail. But here is just an overview and some key anatomic structures. So, like we mentioned, these antibodies are produced by plasma cells. Plasma cells are activated B cells. The structure of it, it's a Y-shaped molecule with variable regions binding to specific antigens, as you can see illustrated here. And as we mentioned, some functions include neutralizing pathogens, something called opsonization, so marking pathogens for destruction, and of course, activating the complement system. But let's dive a little bit deeper into the structure of this antibody. Now, first of all, let's clarify, what are antibodies? Antibodies are also called immunoglobulins. So if you have come across IgA or IgG, some med students think that these are different things when talking about antibodies and talking about immunoglobulins. They are the same. We will talk about why these different types exist in our immunology course, but here you just need to know that there are different types of immunoglobulins. IgA, IgG, IgE, IgM, etc., etc. Now, these are specialized proteins produced indeed by our plasma cells, which are activated B cells, and they are produced in response to the presence of antigens, so foreign molecules or a pathogen like a virus, bacteria, whatever. And their role is they have a key part of the adaptive immune system because they target, like we said, specific pathogens to neutralize or mark them for destruction. So we keep using the word specific because we're talking about the adaptive immune system, which is specific. And therefore, these different types of immunoglobulins come about because they are specific for certain pathogens. Now, what is the structure of this antibody or of these antibodies? They have a very characteristic Y-shaped structure, as you can see illustrated here. And it has two main components that are high yield for you to know. We have a variable region and we have a constant region. So the variable region, we're talking about the tip of the antibodies. And the tips of these Y structured antibodies are unique for each antibody. These regions bind specifically to a particular antigen's epitope. And an antigen's epitope is a small, recognizable part of that antigen. Then we have the constant region, and that's the stem of the Y. And it is less variable and determines the class of the antibody. So when we talk about the class of antibodies like IgA or IgG or IgD, for example, we're talking about something that is defined here at the stem of the antibody. And this region interacts with immune cells and other molecules to carry out different immune responses. And as you can see illustrated here, you see 4 structures. 1, you see 2, 3, and 4. And each antibody, each antibody has 2 heavy chains and 2 light chains, and they are held together by disulfide bonds. So from the point of view of anatomy, remember, variable region, the tip; constant region, the stem. The stem determines the class of antibody. The tip is specifically structured to bind to a particular antigen's epitope and is unique for each antibody. Furthermore, each antibody has 2 heavy chains and 2 light chains, and they are held together by disulfide bonds. And hopefully you will never mix up antibodies and immunoglobulins, knowing that, we're talking about the same thing. When we say antibody, we're talking about the entire structure here. When we say immunoglobulins, we know that we are referencing the stem of the antibody. That will determine which class it is. So it's both the same thing, but it's also a more specific part of that thing. And you don't need to get caught up on listing all of the different classes like IgA, G, IgM, IgE, etc. You will get plenty of that in the immunology course. But we can mention, for example, IgE is a class that is very much involved in allergic reactions and in our defense against parasitic infections, etc. IgG is the most abundant one that we have and provides long-term immunity, and is also the one that crosses the placenta to protect the fetus that is growing. So the differentiation of different types of immunoglobulins and classes has different indications and consequences and where they actually function and what they do. But hopefully now you got a brief overview. Now, continuing, we have different types of adaptive immune responses. So we have the humoral immunity or humoral immunity. This is where B cells are concerned. It involves antibodies produced by the B cell. It is specifically effective against extracellular pathogens like bacteria and toxins. Then we have the cell-mediated immunity connecting with T cells. It involves T cells targeting infected or abnormal cells, and it is effective against intracellular pathogens like viruses. So keep in mind, humoral immunity, B cells, extracellular pathogens. We have cell-mediated immunity, T cells, effective against intracellular pathogens. Now then, immunological memory. So we have the primary response, which is the first exposure to an antigen. This is slower and less intense. If you remember, we differentiated between innate immunity and adaptive immunity, whereby our innate immunity is faster but non-specific. and it also has no memory. However, our adaptive immunity, by its first exposure, is slower, can be delayed up to days, but it will then generate specific responses and with the use of memory cells, remembering the exposure. And when then confronted with a re-exposure, we will have a secondary response, which is much faster and stronger due to these memory B cells and our T cells. This concept is also very important in vaccination because vaccines mimic primary exposure to generate memory cells for future protection. Now, what are some clinical relevancies of adaptive immunity? Well, like we just said, vaccination. We use the memory cells to provide long-term protection. In autoimmune diseases, there can be misguided adaptive response attacks to the body, like in lupus or rheumatoid arthritis. If you mentioned in the introductory lesson, we mentioned that autoimmunity begins when these specific specialized cells target the self, meaning targeting something that's not an invading pathogen, but rather a cell we need and have. And this is where autoimmunity begins. Furthermore, another very important clinical point is in immunodeficiency, so weak or absent adaptive responses, for example, in HIV. And we will dive deeper into these different pathologies relating to immunology. But for example, we talked about the T cell, right? And we differentiated between cytotoxic T cells and helper T cells, CD4 and CD8. When we talk about HIV, the virus is using our CD4 cells to enter and then replicate. And when it does that, it destroys and decreases our CD4 count. And what happens if our T helper cells decline? We become immunodeficient. Furthermore, another important point is allergies. So overactive adaptive immunity to harmless substances will still generate the inflammatory responses we see when we talk about allergies. That can be mild, but they can also be deadly. And moving along, we will talk about hypersensitivity reactions and connecting them to some of the immunoglobulins we mentioned here, like IgE, for example, which is an important and relevant immunoglobulin / antibody in allergies. So in summary then, the adaptive immunity is slow but highly specific and long-lasting. T cells and B cells play key roles in targeting pathogens. Immunological memory forms the basis of vaccination. That's the end of chapter 4. Continue now to chapter 5.